Good afternoon, everybody. Welcome to the second of 3 webinars in the GCOOS Marine Heatwave series. My name is Chris Simonello, and I'm the Outreach and Education Manager for GCOOS. I've had the pleasure of co-organizing this webinar series with my fabulous colleague, Dr. Renata Kamakura. She's come to us from the National Academies as a Science Policy Fellow to GCOOS. She continues to be a tremendous asset to GCOOS, and we're really greatly going to miss her when she ends her fellowship in July. In the meantime, though, she continues to work with our colleagues, Dr. Brindis Wankowski, Zhao Qi, and Deb Rao, to better understand marine heat waves and their impacts on the Gulf region. Like the first webinar in the series, today's presentations are focused on the impacts of marine heat waves on marine species, oysters in particular today. The third webinar in the series is coming July 10th and considers marine heat waves and coastal weather. We hope the presentations make you aware of ongoing work in this space and promote dialogue on how the ability to forecast these events can aid resource management, resilience, and public health and safety. The webinar format will be 10-minute presentations followed by time for questions. And to ask a question or share a comment, you can either raise your hand or type in the question box. We'll unmute everyone following the presentations, but you'll have the option to mute yourself if that's your preference. Now I'd like to hand off to Ren to introduce our speakers. All yours, Ren.
Thanks, Chris, for welcoming us. It's wonderful to see everybody, I guess. Have y'all joining us. As Chris mentioned, for those of you who don't know me, my name is Renata Poulton Kamakura, and I'm here working with GCOOS for the year on marine heat waves. And for those who were not at the first webinar, just to give a really brief overview, basically, we were interested in understanding what the impacts of marine heat waves were in the Gulf for our species, ecosystems, industries, and communities. And what became clear very quickly is that there's some we do know and a lot we don't know. And so this has been a great opportunity for us to pull in some experts, people who are working in kind of a wide variety of fields, but in ways that intersect with marine heat waves, to hear how they're thinking of environmental stressors more generally and marine heat waves specifically as it intersects with their work. So really excited to have a great group of folks talking about oyster related things today. So without further ado, I'm going to introduce our speakers and then let them take it away. So first we have Dr. Andrea Tarnecki, who is an assistant extension professor in the Auburn University School of Fisheries, Aquaculture, and Aquatic Sciences, and is the director of Auburn University Shellfish Lab in Dauphin Island, Alabama. Her research and extension efforts focus on challenges that are identified by the oyster aquaculture industry. Her expertise is in oyster microbiology, specifically as it pertains to oyster health and disease, as well as seafood safety. Today, she will discuss marine heat waves and the off-bottom oyster aquaculture industry, including impacts and mitigation strategies. Next, we have Dr. Jeff Plumlee, who is an assistant professor at Louisiana State University in the School of Renewable Natural Resources and the Fisheries Extension Specialist for Louisiana Sea Grant. Jeff did his undergraduate and graduate studies at Texas A&M Galveston, a PhD in University of North Carolina at Chapel Hill, and a year-long postdoc at the University of South Alabama Dauphin Island Sea Lab. Represent here today. His lab focuses on topics such as fish life history, phenology, and habitat in the context of their management as living resources. And then last but certainly not least, we have Blair Morrison, who is the Science and Monitoring Program Lead for the Mobile Bay National Estuary Program, where she coordinates with stakeholders on the long-term evaluation of ecosystems throughout watersheds of Mobile Bay. As a key component of this work, she helps to oversee the National Estuary Program's Science Advisory Committee and an interdisciplinary group of researchers and scientists experienced in the estuarine environments of southern Alabama. Although her research has taken place over a variety of localities, a wide array of study organisms, her work features the central themes of estuaries, disturbance, water quality, and environmental change. So I think you'll agree, we've got a great group of folks here today to talk about oysters and related things. So without further ado, I'm going to kick it over to Andrea to start us off.
Great. All right. Hi, everyone. Thanks for inviting me here, and thanks, Ren, for that awesome introduction. So I am Andrea Tarnecki, as Ren said, and I'm going to tell you today about marine heat waves and some of the concerns and mitigation strategies for off-bottom oyster aquaculture. So just a little bit of background for any of you who are not familiar with the Auburn University Shellfish Lab, which is where I'm based. We are in southern Alabama on the coast, more specifically located on Dauphin Island on the east end. And we are on the campus of the Dauphin Island Sea Lab. Here on the Gulf side, this is our building here. You can see some footage here on the right. The lab opened in 2003, and we have a variety of research and extension efforts that are focused primarily on oyster aquaculture, but we also work with oyster restoration and other shellfish as well. The mission statement of the lab is to better understand the value of natural environments and promote best practices that lead to sufficient and sustainable shellfish production. This in turn supports harvesters, growers, producers, distributors, and consumers of shellfish. For any of you not familiar with off-bottom oyster aquaculture, this is exactly what it sounds like. It's when you grow the oysters off of the surface or off the bottom of the ocean, so in the water column. This has a number of benefits. It protects the oysters from being buried and smothered in sediments. It protects them from predators. They have access to that photic zone where most of the phytoplankton are growing, so they get a lot more food, they get more oxygen, and they grow really fast. This also makes it easy for the farmers to access the oysters. They can control biofouling, so they can remove any extra setting mussels or barnacles or algae that's settled onto these cages. They also can control shell shape with different farming practices, and this helps to increase uniformity across individual oysters. As you can imagine, this is a lot of labor that's required. And so most of the off-bottom oyster aquaculture industry is focused on providing oysters for that premium half-shell market. Now we're growing them at the surface, which means that we may be more susceptible to heat waves. And so 10 minutes isn't very long, but I'm going to go a little bit over some of the things that we do know and some of the strategies that our farmers can use to help the oysters survive those heat wave events. So the Auburn University Shellfish Lab does have 4 continuous water quality monitoring stations with the locations seen here. So we have one in Grand Bay and one in Portersville Bay. This is where we have 2 oyster research farm sites, and there's also commercial oyster farms in those same areas. We have one over in West Fort Morgan, Alabama, which is a location of 2 other commercial farms. And then we have one in Little Dauphin Bay, which is the bay just north of Dauphin Island. And this serves as a targeted oyster restoration site. So we have a pretty good handle on what's happening in terms of temperature, salinity, and all the other parameters mentioned here. Most of these have been up and running since about 2022, so not a lot of historical data, but we are starting to collect some good patterns. One of the first things that we think of when we think about heat waves and temperature changes is susceptibility of oysters to diseases. Temperatures outside of the comfortable range can suppress immune function and increase oyster susceptibility to disease. The main disease here in the Gulf is Perkinsus marinus, known as dermo for short. And we know that this disease spreads faster, it has higher intensity and higher prevalence when the temperatures are warm, so during those summer months in general, and more specifically during high salinity events, such as when we have droughts. And most dermo cells are released from the oyster host during the day, so again, the temperature is likely to increase the level of dermo infections that we see. Here on the right are some microscopy images of dermo infections. This one at the top, each of these black dots is a dermo cell. This is a very heavy infection that would lead to mortality of the oyster. Here on the bottom is a more light to moderate infection, and the oysters can actually live with that for a while. But aside from the disease that we know about, there is the potential for emerging diseases that could be bacterial, viral, protozoan, fungus. And again, because the temperature stress reduces immune function, it makes them more susceptibility to these. We also see as the oceans warm that some of the tropical diseases can spread northward. So we have the potential to see not only new pathogens, but also ones that are more infective. Of course, oysters eat phytoplankton, and we have been monitoring the phytoplankton in our bays since 2022 to understand what we see and also look at harmful algal blooms. With increased temperatures, harmful algal blooms are expected to increase. Some of these can kill oysters directly. Most of the ones we're concerned about are those that produce toxins that can accumulate in the oysters and have an impact on seafood safety. They can make people very sick if they consume contaminated oysters. But the environmental conditions in general impact the overall phytoplankton community structure, which has an impact on its nutritional value for the oyster. So changes in heat waves can alter that phytoplankton community, impact growth of the oysters and also impact reproductive timing. And we need to be able to reproduce these oysters so we can produce seed for the commercial industry. So these are things that we could be concerned about. Seafood safety, of course, is a big one, especially for off-bottom oyster aquaculture because they are primarily consumed raw. And when we think of seafood safety here in the Gulf, we typically think of bacteria in the genus Vibrio, specifically Vibrio vulnificus and Vibrio parahemolyticus. This little news report from the CDC is from the summer of 2023, where a number of Vibrio vulnificus infections occurred in the eastern US during a heat wave. Most of these were from wound infections, but there were a couple that may have been from consumption of raw shellfish. So we have a project that is empowering oyster farmers to study the Vibrio at their specific farm using these biphasic assays that are shown here. You get results in 24 hours. You take the color change and you can convert that to the number of bacteria per gram of oyster tissue. So we've incorporated this at 2 different commercial farms here in the area, and they can track Vibrio levels over time, understanding the seasonal effects and the effects of farming practices, and just getting ahead to help prevent some seafood safety concerns. So Alabama is really hot, but oysters are surprisingly resilient. What this graph is showing is the blue bars are the average temperature per month for the past 2 years, and the red dots are the maximum temperature during that month. So we do see temperatures in the water that are up to 35 degrees Celsius. Some research out of our lab shows that in those ranges from about 32 to 37 degrees Celsius, the oysters will actually reduce their metabolism in order to conserve energy. This helps them to maintain and survive during these very stressful conditions. So they do have adaptive management strategies within the oysters themselves that help them deal with some of these temperatures. But off-bottom oyster aquaculture is an interesting game because we push those temperature extremes even further with a process called desiccation. I mentioned biofouling. Biofouling is a huge issue in the summer. The bags can get clogged with algae, with barnacles, with setting mussels, setting wild oysters. That reduces the flow in the bag. It reduces the oxygen and amount of food that can get to the oysters. So to get rid of that, we can actually lift the oysters out of the water about every 2 weeks for 18 to 24 hours is typical. And what that does is it exposes that biofouling to the air and it kills that off, but the oysters survive just fine. So what you're seeing here with this graph is a study from summer of 2023. The blue lines are the water temperature and the orange lines are the temperatures in the cages when they were submerged in the water, which is these white spaces, and when they were out of the water, which is the gray spaces. So those are your desiccation periods. And so in desiccation during June of that year, we got those oysters up to about 42 degrees Celsius, which is quite toasty. So again, here's that area where they kind of reduce their metabolism. And up here, right above that 42 degree mark, is where we see them stop reacting to stimuli. So you can kind of poke them and they don't really close. At this point, they're very stressed out, but they can still recover at those temperatures if you get them back to something more within the norm. About a degree higher though, they get to the point where they are dead and there's no recovery. So with desiccation, we do have to be very careful, especially when it comes to marine heat waves. So we have a number of strategies we can use to mitigate these effects. One of the hot research topics now is selective breeding oysters for resilience. We have some programs that are selectively breeding oysters for disease resistance, more specifically for derma resistance, also for resistance to environmental extremes. So mostly salinity right now, but there are a lot of times that selection for salinity can help them be more resilient to these high temperatures. There's a number of forecasting models that exist or are being worked on for harmful algal blooms, oyster diseases, and human diseases that can accumulate in the oysters. So the more warning we have of what might be coming, the more we can prepare to make sure that the oysters are safe for consumption. There are things we can do on the farming side in terms of farming practices. We can submerge the oysters underwater for a period. That allows them to remove or depurate any potential human pathogens like Vibrio that might have accumulated. We can also put them in wet storage facilities to protect them from toxins during harmful algal blooms. So we have a number of options to protect from seafood safety, and these also can help protect the oysters and make their survival higher during heat waves. The good news is we have a lot of, well, good news and challenges, but we have a lot of really great seafood safety regulations, and they're even stricter in the summer. So a lot of the regulations oyster farmers are required to follow already help to mitigate the effects of heat waves. One thing we do on the farming side is we try to avoid handling when there is excessive heat. So this might mean working your oysters in the morning or just not touching them at all while it's too toasty. We can also do that desiccation process overnight or early in the morning to avoid having those oysters out of the water in the hottest part of the day. Managing biofouling is really important. Again, some of that is desiccation, some of it is changing out gear, but that allows more flow to come through, more cooler water, more oxygen, more food. Site selection is really important, looking at the flow in the area, the depth, the mixing, historical temperatures, and having multiple sites can be helpful too, so that if you do have a harmful algal bloom in one spot, maybe you can move your oysters somewhere else. And then there are a number of disaster relief programs available that cover excessive heat, and most of these are available through the USDA. So that was a quick and dirty on marine heat waves and off-bottom oyster aquaculture. My contact information is at the bottom if you want to reach out after this webinar. And thank you very much for your time, and thanks again for having me.
Yeah, thank you so much. That was great. You did a very good job, I think, of covering a lot of ground in 10 minutes. So very much appreciate you
Hopefully I didn't
for
talk too
doing that.
fast.
No, that was great. Absolutely. I want to encourage, you're welcome to keep putting your questions in the questions pane, and we'll always go back and address those in the future. But I did have a question for you, actually, as you were talking. You mentioned, I think, a little bit at the end about selective breeding and some of these kinds of other programs. Are there already kind of sampling in place to look at local adaptation and or any evidence of adaptation over time that's already happened? Or are we saying we don't have the data maybe to understand that yet?
We probably don't have enough data to do it at a fine scale. We do know that there are different populations of oysters in the Gulf that are genetically distinct. And so, for example, in southern Texas, they get really high salinities down there. And so there's a population adapted to that. There's kind of like a northern Gulf genetics, and then there's one for Florida as well. So we know on that scale, but on a finer scale, there are people working on that, but we don't really have that much information on it.
Well, hey, at least we got at least some information and lots of room to grow, which is kind of constantly the theme, I think, in a lot of these. Well, thank you so much. We'll get to you back again at the end, but appreciate you giving us that overview to start.
Yeah, thank you.
With that, I'm going to get ready to pass it over to Jeff Plumlee as our next panelist to do a presentation.
Thank you, Ren. Let's see. Does it look right?
Looks
Okay.
great.
Okay, thank you. So as Andrea did a really excellent job of talking about the really specific mechanisms behind oyster survival and growth and heat, we're going to talk a little bit about a project that we did where we looked at heat waves as an indicator of oyster population health from the fisheries perspective, or so from the wild harvested perspective. So we know that oysters in the Gulf of Mexico are incredibly valuable, but the fishery has been on the decline historically from its historic peak in the 1950s all the way through to now, where we saw a historic low in Alabama oyster landings in 2014 to 2018, where it was a fraction of what it has been historically. Oops. There we go. And like I said, oysters are as valuable as they've ever been. So our motivation for understanding exactly why those landings are low and what we can do to mitigate the different environmental disturbances that may drive those low landings is at the all-time high. So we're really, really motivated to be able to figure out what exactly is going on with oysters. And what we found through some preliminary analysis looking at what this kind of fishery decline was that, you know, concurrent to all these declines was heat waves and extreme heat and increasing temperature. We noticed that, and that's pretty pervasive across the country, especially in the Gulf region. We noted that that was a pretty strong correlation between declining fishery and extreme heat. So we decided to look into a little bit deeper. For this project, what we did was we defined heat waves, and we defined them as 3 continuous days where the daily maximum temperature exceeded the 90th percentile. Then the 90th percentile average temperatures in July and August. So our hottest of hottest times. And what we know about oysters is why heat is probably a good indicator of population health is that their life histories are really tied to temperature. So let's say, for example, oyster spawning occurs, and we can, whenever you see a greater than 2 degree decrease in temperature within 24 hours. You pretty much set your watch to it. Wild oysters in Mobile Bay will be spawning if you hit that condition. And then also what we know is that their larval duration, so after they've been fertilized and they turn into larvae, that larval duration before they settle on structure is also temperature driven. So the hotter the temperature is, the faster the larval will settle onto substrate. The colder it is, the longer the larvae will take. And to give you a little bit of indicate or to kind of point why we're using this specific life history stage, so the post-larval life history stage, is because often in fisheries, what we're trying to do is we're trying to look at recruitment. So how much new biomass is being added to the fishery in a given year. And so the new indicator of biomass for oysters is oyster spat. So the small post-larval oysters. And recruitment is kind of the holy grail of fisheries. We're always trying to figure out what the mechanisms of recruitment are and how we can predict recruitment. And good recruitment is really difficult to predict because in the case of good recruitment, good fishery recruitment, you have to have a lot of things that go right at one time. A lot of food availability, habitat availability, a lot of spawning success. That all has to work in conjunction with a suitable habitat to have good recruitment. But bad recruitment only really needs one thing to happen for it to occur. So for this project, what we were looking at indicators of poor recruitment. Okay, so can we find a reliable indicator that shows us that oysters will be in, or early new biomass will be in low abundances in a year, and so that kind of led us back to heat. And these figures right here aren't especially intuitive, but what we're trying to look for is the B example, where in extreme conditions, so on the left side of that vertical line, and how often can we predict poor recruitment? Okay. Now, we said we defined heat waves earlier as 3 continuous days where you're exceeding the 90th percentile of temperatures. That's a normal heat wave. Like Andrea said, heat waves are normal in the northern the northern Gulf region. They are we're dealing with heat all the time. But the extreme conditions, the 90th percentile of the 90th percentile, those are the things that we're really interested in. And those we had to kind of create cutoffs for. So in this project, we looked at not only the Mobile Bay fishery, Mobile Bay oyster fishery, but also the Apalachicola Bay oyster fishery, because both are historically really abundant fisheries. In Apalachicola has seen similar declines. In fact, it's resulted in oyster fishery closures in that region. So similarly, we're betting on the fact that there's the mechanisms that are driving this heat-related changes to population dynamics and mortality are the same in both regions. So, but for both regions, we have to create these kind of cutoff metrics. So what is a extreme heat? How do we define extreme heat waves in these different regions? So we looked at the kind of the four components of a heat wave. We looked at magnitude, which was at maximum yearly temperature. We looked at the duration, so the annual number of days that this extreme heat occurred, and then the consecutive number of days that were this extreme heat occurred. So if it was, so for example, exceeded this 11-day threshold, that was what we consider to be an extreme heat wave for duration. And then the frequency of heat waves. So how many heat waves occurred in a given year? So that cutoff was 6 and 5, respectively, to the different regions. So to go back to that original concept, how in these extreme conditions, so under extreme heat waves, how likely was it that we had poor recruitment of oysters? So this figure that we have here is well described on the far left side. You've got on your vertical line, you have extreme conditions and normal conditions. Extreme conditions are on the right in our figure. And then below the horizontal line, you have good recruitment versus poor recruitment. So above median recruitment or below median recruitment. What we found was that for all the metrics and the components of these heat waves that we're looking at, duration, so the duration of heat waves was the most influential variable that we had to predicting recruitment of oysters. So in both Mobile Bay and Apalachicola Bay, if you had greater than 11 consecutive days above that heat wave threshold, you had poor recruitment in Mobile Bay 80% of the time, and you had poor recruitment in Apalachicola Bay 88.9% of the time. So kind of a smoking gun here. So it was really interesting that this was so so tightly correlated with the recruitment of wild oysters, this heat wave metric. But like Andrea was saying before, it is not a one-to-one comparison. It's not a one-to-one indicator. You know, there are all these different mechanisms that happen at the same time as extreme heat does. You have increased predation, decreased available oxygen. Potentially, if you have low salinity at the same time you've got heat waves, you've got increased mortality, metabolic stress, physiological stress. So while this is a really strong and powerful indicator of poor recruitment, it's not a mechanism, but it is. That's kind of what was being used for right now. We're saying in these years where we have these extreme heat conditions, let's take a second look at the recruitment indices for these for oysters and set our and which can lead to setting new regulations, setting different catch quotas and all that stuff. So feel free to take a look. It's been published in a journal called Ecological Indicators, I believe. Yep. And happy to have you look at it. And if you've got any questions, please forward along, or any sort of other critters that you'd like to look at, or recruitment indices that you'd like to look at that correlate with heat, like we assumed oysters would. So yeah, thank you very much.
Thanks so much, Jeff. Yeah, I thought that built quite well off of the kind of base that Andrea really built for us, diving deep into this one example, which is awesome. I know we have a question in the questions pane. I'm going to come back to it in the discussion because I know that was related to Andrea's presentation as well as Jeff's, which is awesome. So I guess one question that I might add here is, I know you're talking about how it's much easier to predict poor recruitment, which makes a lot of sense because there's one thing has to go wrong. I'm curious, for those one on the graph that you showed, there's a couple examples where even though there were extreme conditions, they did really, really well. Do you think you'd ever go back and be like, I wonder why, despite the fact that you're in some extreme conditions, these couple oysters seem to handle it, if there's some kind of enabling condition that helps them do well? I don't know if that's something you'd ever be interested in.
Yes, absolutely. I think the whole under being able to predict the actual instead of having this threshold effect, if we were able to do this one-to-one prediction, that's the dream. But there's just so many things that that can go right. They have to go right for it to go well, or it just really takes one thing. And I think that was the most telling thing that we saw here, is that it is really clear that it was the consecutive heat wave days is a really good indicator of poor recruitment of oysters.
Yeah, absolutely. I was going to say, in biological sciences, I rarely see a 90% or 80% anything. That is not usually what shows up. I'm used to.
Exactly. Yes, exactly.
Well, awesome. Thank you so much. We're going to come back to ask you some more questions in the final discussion, but thank you so much, Jeff. That was awesome.
Yep.
With that, I'm going to pass it over to our last presenter, Blair, to finish us out before we go into our panel discussion. So over to you, Blair.
All right. Can you see my slides? Beautiful. Love when the tech works. All right, y'all. So we're going to take a little bit of a detour from the empirical science side and start talking about policy applications and recommendations for management. So my presentation today is going to be on perceptions of stress on oysters, and these are insights from the Mobile Bay NEP's stressor matrix evaluation. I'll get into more of what that means and what the outcomes of that are. But first, I want to give folks just a brief introduction into who we are if you're unfamiliar with the National Estuary Programs. So the Mobile Bay NEP, we're one of 28 national estuary programs in the United States. We are funded by the US EPA, but we also are supported by the state of Alabama, local governments, and the private sector. We are a division of the Dauphin Island Sea Lab, so shout out to Andrea. Woo, we love our Sea Lab buddies. We were established in 1995, and we occupy this really interesting space of being a non-regulatory and non-advocacy organization. So that allows us to really be a place-based program, and our main charge is protecting and restoring water quality and ecological integrity of estuaries of national significance. We were created through amendments to the Clean Water Act in 1987, and really we function as a collective action organization. Overall, that means that we work to create consensus on resource management for our local estuary by bringing together all sectors of our community to come to the table and identify where environmental issues are and what the next steps are to address them. And on our map here, we're number 19, where this little orange star is. So one of the tools that we use for collective action is the stressor matrix or the stressor evaluation. I'll use those interchangeably. They're all the same thing. But this is a consensus building exercise that has been used to identify areas of environmental concern, and we use it to prioritize broad resource management actions within the estuary. And this was developed by our Science Advisory Committee within the NEP. And this exercise is used to engage both the members of that committee and then the broader resource and research and resource management community to kind of weigh in on areas of stress. And this is completed roughly every 10 years or so, but the tool itself has the potential to be used during emerging events as a reactive assessment tool. But generally, we have decadal participation for this evaluation. It's important to note that this is kind of a wisdom of the crowds assessment. So these are perceptions. They're not necessarily empirical, but they are based on the informed opinions and perceptions of members of our local research and resource management community. So they're still informed, opinions. And a lot of times we will gut check those findings of this evaluation with what we do know about the estuary. And overall, the evaluation consists of 11 local habitat types, 12 important ecosystem services, and 13 stressor categories. So the 11 habitats that are part of the stressor evaluation, they span across our service area from uplands to offshore, and they include freshwater wetlands, beaches and dunes, intertidal marshes and flats, oyster reefs, of course, that's the one that we're all here for today, streams and rivers, submerged aquatic vegetation, subtidal habitats, riparian buffers, pine savanna forest, longleaf pine habitat, and maritime forest. So lots of different habitats across the coastal landscape. And then also across each of these habitats, they each provide a suite of ecosystem services. And those include things like biodiversity, fisheries habitat, sediment and nutrient retention, water quality enhancement, wildlife habitat, carbon sequestration, nesting habitat for birds and turtles, storm buffering and hazard protection, flood control, groundwater replenishment, oyster production, and primary production. All of the important ecosystem services that we need our coastal habitats to provide. And then again, across this coastal landscape, we have myriad stressors that affect these habitats and their ability to provide those ecosystem services. And these are the 13 stressors. So we have land use change, climate variability, fragmentation, sea level rise, dredging and filling, sedimentation, chemical contamination, fire suppression, invasive and exotic species, nutrient enrichment, pathogens, freshwater discharge, and resource extraction. All in all, that is over 1,700 combinations of factors, which is a lot. And I know our science advisory committee, when they were originally developing this exercise, they're like, can we boil it down any more to kind of make that a little less cumbersome? And it turns out, no, you can't, because there's so many things to consider. So overall, how do we get people to give input on the combinations of those factors? So for the stressor evaluation, our participants are asked to score each combination of factors. So within a habitat, the ecosystem service being affected by a stressor. Each combination, they score from 0 to 3. A score of 0 is categorically saying that that stressor imparts no or very minimal impact on the ecosystem service provided by that habitat. Whereas a score of 3, it means that a stressor imparts a large negative impact. And basically anything in between is kind of the shades between those two extremes. And we asked our participants to leave blank any combinations that they're unfamiliar with or unsure of, because again, we want kind of the best educated opinions on what the impacts of these stressors are. So the median values of all responses are calculated for each combination once we get everybody's input. And we said that a median of 2 or greater is the threshold indicating a higher perception of stress for any combination. Originally, the evaluation was a matrix and an Excel sheet, which you see on the left, which, as you can imagine, was incredibly cumbersome. Getting participation in that was a huge ask. So in 2023, we upgraded this to be more user-friendly, and we migrated the whole thing over into Mentimeter, where instead of filling out little cells, you got to do little slider bars, but it still preserved all of the same data on the back end. So for example, we'll just walk through this really quickly. As I mentioned, the stressor evaluation, you're evaluating the effect of a stressor on a certain habitat's ability to provide an ecosystem service. So in this example, we're talking about pine savannas. We know that fire suppression has a strong negative effect on biodiversity because many pine savanna species are fire dependent. So we give that a 3. So we know that fire suppression greatly affects the biodiversity of a pine savanna. We also know that land use change is rapidly converting pine savannas to residential or urban areas. Climate variability has impacts on many of those species in pine savannas, and pine timber is harvested often. So that's kind of the rationale for each of those impact kind of scores that we've given. For fisheries habitat, that's not really a common ecosystem service provided by pine savannas. So the combination scores for that are really low. And for this one that's blank, let's say this person who is filling this out isn't really sure what the effects of climate variability are on nesting habitat, so they left it blank. So moving into why we're all here today, we're talking about oysters. This table is a little busy, so I'm going to walk us all through. So this table is showing the median response values for all combinations of ecosystem service and stressors within the oyster reef habitat. So we have our ecosystem services on the left column, and the stressor categories are along the top. The warmer or the more yellow a cell is, that indicates a higher perception of stress. So within oyster reefs, freshwater discharge, dredging and filling, sedimentation, and climate variability were the greatest identified stressors. That means they had the highest number of medians that were greater than or equal to 2. And the ecosystem services under the greatest stress in this habitat include oyster production, of course, biodiversity, fisheries habitat, and water quality enhancement. And overall, the medians that were developed or that were calculated for this habitat type were determined by a minimum of 8 responses. So at least 8 people filled out all of these cells, and some of them had a maximum of 16. So it's also important to note that the highest medians calculated for this habitat were related to the habitat's ability to produce oysters. So this is in the purple box, this 2.5 and this 3. And that means that freshwater discharge and climate variability were the greatest perceived threat to the ecosystem service of providing oysters. To gut check this, we can consult some background information about the estuary and the ongoing research into long-term trends and hydrologic conditions. Even though we just work solely in these kind of coastal counties here along coastal Alabama, we have a really big watershed. And we know that the kind of freshwater events that we're seeing locally are increasing in intensity. So again, freshwater discharge, it makes sense that that's becoming a greater kind of stressor that people are perceiving. And also we've seen that temperatures in the bay, in Mobile Bay, have raised by anywhere between 0.4 and 1 degree per decade. And those have been recorded via our real-time coastal observing system stations. There's been a lot of extreme temperature fluctuations in recent years and lots of hurricanes as well. So plenty of things that are stressing our oysters. But all in all, the perceptions appear to be fairly aligned with what's currently known about the estuary. And really quickly, we're going to zoom out to the stressor matrix as a whole. So we have our top rated habitats, ecosystem services, and stressors that came out. And it's important to note that oyster reefs weren't among the top habitats, nor oyster production one of the top ecosystem services that came out from this study. However, many of the stressors that scored highly in the oyster reef section are highly ranked stressors overall across the local coastal landscape. But this doesn't mean that oyster reefs aren't under stress or perceived as being safe. In fact, perceptions of stress on oyster production rose in our most recent evaluation as compared to the last time this was done in 2012. And this is kind of in that orange box. These pink cells show medians that increased over that 10-year period. So of course, we're kind of seeing more stress or more perceived stress in the ecosystem service of oyster production. And also our stressors of sea level rise and climate variability, again, leading into our marine heat waves, have higher perceptions of stress across all habitats. And this is important because as a resource management kind of consensus agency, our the current perceptions of stressors and the shifts in perceptions of stress need to be incorporated into our monitoring and management strategies. We're doing this by talking about changing landscapes and changing climate in our next comprehensive conservation and management plan. And what this means is that some of the goals that are in our next 10-year plan really affect oysters. So our goals are to reduce upstream sediment loading from local tributaries by 30% by 2035. Also establishing nutrient criteria for coastal basins by 2030. and increasing the number of communities and businesses investing in improvements to environmental quality, namely improving marketability and sustainability of oyster aquaculture. And kind of in the background, using the stressor evaluation as a tool moving forward, our science advisory committee is really interested in looking at what future iterations of this stressor matrix, what they'll look like, how often should we be completing this? Because maybe 10 years is not enough. Maybe we need to start doing this on a 5-year basis because things are changing so quickly. And then also ensuring that we have a wide diversity of participants who are bringing expertise from lots of disciplines and expertises. And with that, happy to answer any questions, and thank you to everyone for listening.
Awesome. Thanks so much, Blair. Yeah, I think that was a great way to kind of wrap it up, looking how at least some management related things can be responding to the stuff that both Andrea and Jeff brought up. Since we are at our kind of final presentation and into our discussion mode, I'm going to invite Andrea and Jeff, if you would like to come back out and answer some questions from the group. I know we've got a couple that have already been posed, which is great. I'm going to attempt to allow some people to unmute, but I'm not sure it'll let you. So I'm going to hit unmute all participants. And that way, if you want to unmute, I believe you'll be allowed to. So I know we have a question, Enrique. I think you asked a question in the chat. I don't know if you are around and want to unmute yourself to ask it, but you should maybe be able to. I'm going to give you a second in case. Okay, it seems to not actually be unmuting you, for which I apologize, but it's a great question that's in the chat, so I'm going to ask it on your behalf. So thank you for posing it. And I'm going to encourage other people to pose your questions in the chat to make sure that we can see them and ask them for you. So the question from Enrique is there evidence of hypoxia induced by heat waves that it has affected oyster farming? Any of y'all know anything about that?
Yes, so we do see some incidences of hypoxia, sometimes related to heat waves, sometimes not necessarily. The heat waves can also lead to algal blooms, whether they're harmful algae or just normal algae, and they can, use up all the oxygen and lead to hypoxic events. So we do see some of that. For the off-bottom oyster aquaculture industry, it's not as often as you would see it maybe on the bottom, because they are right there at the surface, so they're getting some wave action and getting some mixing there in that strategy that you may not see with the on bottom farming. But there is evidence of that, yes, for sure.
Yeah, thank you. Great answer. Another question we have from Michael in the chat is, this is kind of great, I've heard this rule as well, used to the basic rule that you should only eat oysters in months that contain an R. Is that valid, or are they safe to eat anytime? What are y'all's takes on this rule?
I can jump in first. So part of that actually comes from the fact that oysters are spawning in the summer. So when oysters spawn, they get really thin and watery. Believe it or not, when you're eating a raw oyster, if you got a nice fat one, a lot of what you're eating is gonad. It's egg and sperm. So there is part of that is part of why that that saying came around, is because in the summer, some of the oysters can be really thin and watery, and we have, I don't want to go into all the ploidy stuff, but we do have oysters that do stay fat and plump year round. In terms of the seafood safety side of that, that's because, of course, the Vibrio bacteria that are naturally present in the marine environments do like the warmer weather. They're in higher abundance during that time. I do want to say that just because it's within that species of bacteria doesn't mean that it's pathogenic. There's certain genetics that lead to that. It seems to be kind of regional, but there are really strict regulations that are known as time to temperature requirements that are set forth through the National Shellfish Sanitation Program, which regulates interstate shellfish transportation in the United States. Those requirements are basically, when you get them out of the water, you have a certain amount of time to get them down to a cold temperature to make sure that those bacteria are not proliferating. And those requirements get shorter and shorter as the temperatures get warmer and warmer. So of course, I would never I would always say, you know, talk to your doctor if you have concerns. But for most people, raw oyster consumption in the summer should be fine.
Thanks for that. Yeah, it was very helpful. I definitely also could have, yeah, I had also heard that rule, so that's helpful overview for me for sure. I have another question that I think is directed towards Blair, which is what group of people were invited to answer the stressor evaluation survey?
Yeah, that's a really great question. So of course, all of the members of our science advisory committee, these are all folks who have usually been involved with the kind of the creation or the maintenance of the National Estuary Program. It's like I'm pretty sure Andrea joins on occasion, and then Jeff has been involved with us through some of the decadal study work that he's been a part of. But really, it's a combination of folks at the Dauphin Island Sea Lab, lots of professors from local institutions, also our like research scientists and scientists that are in our state and federal agencies, local resource managers are part of that. We have folks who are on the environmental side of some of our local engineering firms. It's really a pretty broad kind of suite of people that bring a lot of different flavors of expertise to the table. But typically, whenever we do a stressor matrix or stressor evaluation, we try to get folks who can kind of fill in the gaps if we know that there are certain topics that our current kind of core group of people can't really speak to. And then this past time that we did the stressor evaluation, we also wanted to make sure that we had a good contingent of folks who responded to it the first time back in like 2010, 2012 to see how those perceptions have changed over time. Of course, that could be challenging sometimes because people retire or they move on. And then in that case, contemporaries for them were identified.
Thank you. Yeah, that's an interesting overview. Trying to pull from a broad range of people. Okay, I have a question for y'all, actually. That's maybe because I do a lot of data related things, so shameless plug for GCOOS. But I'm curious, when you're talking to folks who are making management decisions, some of you I know make those kinds of decisions yourselves, what sorts of data sources are people using? Like when we think about, I mean, as you mentioned, there are oysters that can be impacted by a bunch of different things. Where are they getting their info for, you know, the degree of phytoplankton blooms or temperature or salinity or anything else? And this is open to all of you because I think all of you have at least some experience with these kinds of things.
I'll say on the resource management side, we pull from a lot of different data sources. Typically, it's like a lot of the time we kind of resort to the state data, like the regulatory kind of agencies and the data that they collect, because they oftentimes have the final say on a lot of things. But also I would say for like temperature and just kind of the basic hydrographic conditions, coastal Alabama has a really fantastic array of sensors, the ARCO stations, so the Alabama Real-Time Coastal Observing System. That's a great resource for both resource managers, researchers, and just the general public to see what's going on. As far as phytoplankton goes, I know that the Department of Public Health has a pretty good monitoring program for specific genera of harmful algae, and that's a great resource as well. Sometimes it can be a little hard to find their data, but if you know the right person to email, you can get more data than you'll know what to do with.
Yeah, that was great. I don't know, Jeff or Andrew, if there's anything you would add.
I was going to jump in with another question if they don't want to comment or weigh in on that. So Andrew, you had mentioned, I was pretty fascinated by the comment that you made that when temperatures get really extreme for the oysters, they actually decrease their metabolism, which kind of goes against all of the enzymatic reactions and functions that as temperature increases, rates of reactions increase. So are you seeing like loss of function in like core systems when they're getting to that point?
So they do, as you said, as temperature increases, generally their metabolism also increases, but they hit a threshold where, you know, upwards of a certain temperature, they go into kind of a resting metabolic rate. And so we haven't really taken a deep dive into the specific areas that we see them, kind of suppressing. But we do know that, they'll hit that kind of metabolic peak. And then, so these have been, I should say, from acute experiments. So we've been ramping the temperatures, over time and continuing to heat them up. So we don't have the data necessarily of past when the temperature starts to go back down. So there's still a lot of questions there, but we do know that they are able to, in some way, alter their metabolism to help them resist the extreme stressful temperatures.
I wonder if anybody's done any of like the heat shock protein studies to see like, it's fascinating to hear you talk about that and how they adapt. Thank you.
Yeah, of course.
Jeff, did you have your hand up for a second? I thought I saw you.
Just wanted to put a pin into what you were saying about where we're getting our data. I think it's exceptionally valuable. The state collected data, the federally collected data, it all goes into answering these questions. The state of Alabama does a fantastic job of monitoring their living marine resources and have done for decades. I mean, I kind of, think about it when I'm doing analysis with that data is that these are pension projects. Somebody spent their entire careers collecting these very specific data points, enough so that they could collect a pension at the end of it. It's just remarkable to think about the effort that goes into it and the forethought that people had to taking and recording that data and keeping it in a way that is so useful for us all to answer these types of questions.
Yeah, I'm glad you brought that up. I think Andrea, you mentioned this as well, is we're getting a lot of new data sets which really help us go in depth onto some questions, which is awesome. And to understand things like heat waves, you have to understand what the normal temperatures are for a given area, or even know what the normal demographic change is in a given area. And for that, you need really long-term data sets, these kind of pension data sets that Jeff just really pointed to. And it's super, super key for us to be able to have that, to be able to understand some of these questions. So I'm glad you brought that up. I know we are very close to time, so I want to say thank you so much to all of our panelists. Y'all are great. Great excuse to get to chat with y'all a little bit. It's been wonderful. And then I will kick it over to Chris to actually fully close us out.
Thanks, Ren. And thanks to Andrea, Jeff, and Blair, and to everybody for joining us today. You can hear these talks, and if you've missed any others in the series, they're available on the GCOOS YouTube channel, and we'll circulate that link to all of the participants after the webinar. Please join us July 10th for the final webinar in the series, Marine Heat Waves and Coastal Weather. We'll be hearing from Brian Dzwonkowski, who is an important operator of the ARCO site that you mentioned and a longtime GCOOS data node. So we're happy to hear that that's being used in that capacity. He's from the University of South Alabama and the Dauphin Island Sea Lab. We'll also be hearing from Sean Milrad from Embry-Riddle Aeronautical University and from Coral Lozada from the Houston Methodist Academic Institute. Until then, thanks to everybody and be well. We'll see you soon.